everyone. Many of us who are currently abroad and getting trained in different research fields dream of returning to India and contribute to the growth of the scientific ecosystem there. A job in an academic institute that focuses on research and training is an ideal position that many seek. But as you all know, getting such a position is very challenging. One of the most important hurdles in this transition process is to become scientifically independent from our mentors and readiness to start an independent research lab. This readiness has to be demonstrated through research proposals. Many questions come to mind when we are at this juncture. How do we identify a unique research problem to work on? How do we know if this problem is relevant or the methodology that we propose is appropriate? Most importantly, we also need to know if this question is appropriate in the Indian context and if there is infrastructure available to do the work. We at Sci-ROI have taken multiple initiatives to help our members to make transitions to India. To better understand this aspect of research proposals, we are now interviewing faculties who had already transitioned to India. We will try to understand through their experience how they identified an interesting research problem to work on that was independent from their mentors. What were the challenges that they faced when they tried to execute this idea? We will also find out how they tailored their initial idea to suit the unique strengths and challenges in Indian scientific ecosystem. Today, we have Professor Vidita Vaidya, a neuroscientist at the Tata Institute of Fundamental Research in Mumbai. She joined the Department of Biological Sciences in March 2000. Her lab is interested in understanding the neural circuitry of emotions, particularly on how life experiences modulate the neural circuitry and the alteration in the emotional neural circuitry that underlies complex psychiatric disorders like depression. Dr. Vaidya received multiple awards, such as National Bioscientist Award in 2012 and Shanti Swarup Bhatnagar Award in 2015. She's also a fellow of the Indian National Science Academy and the Indian Academy of Sciences. Most importantly, Dr. Vaidya is an excellent mentor and received the Nature Award for Mentoring in Science in 2019. And according to sources, she is an avid storyteller. Today, let us hear from Dr. Vaidya how she started her career as an independent investigator. Professor Vaidya, please tell us a bit about your background in science. What got you interested in a career in science?
Sure, Atul, and please do call me Vidita. We have a nice long chat here planned, so it'd be good if you call me that. Okay, so I come from a family where there have been scientists, and actually it's a family with my parents are both MD PhDs. My uncle's a malarial parasitologist. So I can't say I have a fairly privileged and lucky background in that I already had access to, through my parents and through my uncle, I knew what the research ecosystem was to some extent already. And as a child growing up, my grandfather's a novelist, a writer, and a freedom fighter. So there was this other aspect, which is storytelling. And as you said, storytelling is one of those things, right? So my grandfather, my great uncle is a poet, my great aunt is a poet. So there was these two streams that were there, which was literature writing and, novelists. And then the other side was doctors, medical doctors in science. And so as a kid growing up, how it used to be very narrow in those days growing up. It was like either you do medicine or engineering if you do the science streams. And I knew I didn't want to do medicine. I knew I didn't want to do engineering, but I still knew I wanted to do science. So, it was this broad sense of ended up doing life science and biochemistry for my undergraduate. And we had one course in neuroscience during that time, but that just captured my imagination that it was this interim space between psychology, neuroscience, behavior, and yet there were fairly incisive molecular cellular tools also. So you could use very cutting edge tools, but the questions were these amazing questions, right? So as a kid, as an undergraduate, I just thought this was amazing, but I just didn't have any real neuroscience exposure. So right after my undergrad, I went to the US to do my PhD. And I remember in the first semester thinking, how will I even survive this? Because I've not even really done any basic neuroscience so far. So I took an undergrad neuroscience course while doing my PhD, and that was very useful. And then by then I knew that behavioral neuroscience is what I was interested in. And that hasn't changed ever since the first year of my PhD. That hasn't changed. Behavioral neuroscience is what I find the most fascinating. And I started working with rodent models. I still work with rodent models. So that hasn't changed either. Though along the way there have been, we've gone off the track that perhaps I started off with, many meanderings. In a sense, one of the luxuries that India affords you is the ability to meander. And I think that is an asset that you can have when you're in Indian science. So I've definitely taken advantage of some meanderings for sure. Yeah.
Nice. So Vidita, now your lab actually works in the neural circuitry of emotions. So how did you come across this theme of research for your lab?
So I worked with one of the leaders in the field for my PhD. I worked with Professor Ronald Duman at Yale. He was my thesis advisor. And he really set up in many ways the community or the field where people were looking at neurotrophic and neurogenic changes in the brain that took place in animal models of depression, and also were targeted by pharmacological as well as other antidepressant treatments. So in a sense, I would say that very early on in my PhD, I got interested in circuits in the brain that are associated with mood disorder dysfunction, and also that are targeted by drugs that are used to treat depression or anxiety. So that broad framework was already there early on in my PhD. And even in some sense, in my postdoc, I continued asking questions, perhaps more about neurohormonal mechanisms. When I came back to my lab, I was particularly interested in sort of the persistence of behavioral changes that arise in response to life experience. So a lot of what I had done in my PhD was in adult animals, where we were looking at changes evoked in either models of stress, which were initiated in adulthood, or drug treatments that were also given in adulthood. And in my own lab, I decided initially to focus on early life, that early critical period window of postnatal life, which can then have, if there is trauma or stress or adversity in that window, and we know this from clinical research, the effects are very long lasting and very often persistent. And I guess by then I was very interested in why is it something so unique about those postnatal developmental windows that can result in such persistent, long-lasting changes. And so that's what I initially started off with in the lab. And then along the way, I worked with serotonergic psychedelics as a PhD student also, but my lab has really focused on how serotonergic psychedelics influence sort of the neurotrophic milieu in the brain, and also how they bring about sort of bioenergetic changes by targeting mitochondria. So this is something that we've also been doing in my lab. Yeah. So those were, they were not what I was working on at all at that point in time, but right. I think the broad framework of the area is fairly similar to what I started off with in my PhD. Yeah.
So this kind of comes into the next question in some ways. So it looks like your initial idea was kind of an extension of your PhD work in some sense. Can you give us a little bit more insight into the specific research proposal that you gave to TIFR while you were applying for a faculty position? I know it's been quite a long time ago.
A long time ago. It's also sometimes I wonder, I was a 29-year-old kid when I started, Atul. I think they just took a leap of faith with me and gave me a chance. Really, it's not like if I look back at my own research proposal, no, it was not as honed and as well written and put together as perhaps some of the research proposals I see from faculty applicants today. I had just done a 2-year short postdoc by the time I was ready to come back, and I really wanted to come home, and I was just 29. And I think it was like literally a leap of faith, right? So they decided, I came, gave a talk, and I gave a sense of the broad questions I was interested in. And I was certainly interested in sort of neurotransmitter, neurohormonal pathways that can influence these circuits in the brain that drive mood and anxiety related behavior. That was one broad area. And I was very interested in looking at the effects of early trauma from a lifespan perspective, which perhaps had not as extensively been done. That definitely had been looked at, but early trauma, more often than not, the studies were in young adulthood. So soon after early trauma. I wanted to ask whether the consequences would vary if you looked across the entire lifespan. And I thought that that was a niche advantage I could capitalize on in India, partly because one, we don't pay animal costs on the scale that the US does. So I could imagine doing these sort of more longer term studies where I could look at early adversity and let animals survive across the entire lifespan.
Right.
And so that's certainly a competitive advantage we have in India, because that's something that is possible. So I was also looking for a niche where I could do something which might give me some degree of competitive advantage. And more than that, I was just saying, oh, I mean, what is it that I'm going to be able to do back here in India that is going to, allow me to at least get my lab off the ground, publish, be able to attract funding. I think I was just too excited that they were even willing to take a chance on me at that time. So if you ask me how well my proposal was written, it was terrible. It was just, it was a little bit of a mishmash of ideas. But I think whether it's a well-written proposal or not, I think what people look for is someone who's going to come in with a certain degree of energy to put in the effort to build something, especially if you're coming into a space which doesn't have everything already laid out for you, right? So there's a fair bit of that pioneer effect of, okay, you come in, then you have to build stuff, you actually have to get things off the ground, and you shouldn't be daunted by that. I mean, that's actually one of the fun things about doing things in India. It can be challenging. I won't paint only a sort of a Pollyanna version of it, but it can also be really interesting because, sometimes you start looking at a problem slightly differently because you have to now look at trying to solve it a slightly different way. So I think people are looking for a combination of well-written proposals that indicate that this person has thought through what they can actually achieve in 5 years, and that there's a realistic sense of what it will take to get off the ground in the ecosystem you're entering. I mean, it will vary across India. Like, there are institutions that are very young, just starting up. There are institutions that are far more mature and have had now years and years of people working. Also, the area that you're coming in on, so that there was hardly any vertebrate neuroscience at TIFR when I came here to the Mumbai campus. So there was always going to be the need to sort of, roll up your sleeves and get in and actually do stuff. So yeah. So I know it's not a very well thought out answer, but it's truly 21 years now. So
So it also kinds of, answered a little bit of my next question, which is like, you had a different kind of infrastructure and a different kind of experience doing it and doing your research for your PhD and your postdoc abroad. But now you're coming to India where the things are very different. So your idea had to be molded to kind of, to suit the Indian system somehow. Can you give us a little bit of an idea, like, how you had to go through that process?
Yeah, so that was interesting now that you're asking me that. When I came back, we barely had an animal house with barely any animal facilities. We had to first hire a veterinarian, right? We had to find a vet who would come to TIFR and work with us. And then we had to find, in fact, me and one of my students went in. So now we come to the storytelling part of life, which is one of my first master student. I actually went to Hafkin to buy some rats and bring them back, and brought them back in my car. And the trunk of my car, we had to leave the boot open because the animals were in a cage. And the taxi driver driving right next to us in the road, and he kept looking at us. And then at some point there was a signal, and he comes to the signal next to me and says, "Madam, aapko pata hai na ki aapke, in the boot there are rats." And it was just a surreal conversation. But we had to get our first batch of animals in from Hafkin, second batch of animals in from Bangalore, figure out which is a cabin, which is pressurized that can bring in animals in the plane. There was a whole bunch of things we had to figure out, but it's interesting. I think there's an interesting element to discovering these sorts of things also. You end up talking to a whole bunch of people. Sometimes things are really difficult to solve. In fact, they take a long time. Then you have to figure out a way around the problem. But overall, I think you sign on for that depending on, you go in with your eyes wide open. And I knew I was coming to an institute in which at that point in time, there were no vertebrate biologists. There was one recruit before me, and it was literally two of us at that point in time, a year apart from each other, and both vertebrate biologists. So I knew that it was not going to have a setup because I'd come here, I had interviewed, I had seen the place. So I was eyes wide open, right? So I also knew that meant that in the first 2 years, I will have to figure out ways to stay productive because how am I mean, students are going to start coming. You can't then tell students, oh, the whole of 2 years is going to be going and bringing some animal from here or doing something. That's not viable, right? So you have to come up with strategies of how will you solve such that you can get off the ground at the same time as allowing yourself to build. So that's a toggle, right? You kind of have to figure out, and you need to publish relatively soon because you also need to attract funding. I mean, eventually people do give you funding on promise, but at some point they also want to see actualization of whatever promise it is that you. So I think, being able to look at what's your short term, what's your medium term, and sort of long term, and then really making sure that you don't divert your attention to only one of those three, right? If you are so focused on the short term, you won't build anything which is longer term more exciting. I mean, your high risk, high reward projects may take time. You know that those are going to take time and they're going to take effort. They're going to take a whole bunch of things being set up. So, but you shouldn't give up on those. But at the same time, you better have some low hanging fruit that you can go after so that you can toggle between these three. And your first batch of students have stuff to do in the lab. You're in the lab with them too. I mean, you're physically in the lab doing experiments. So yeah, I mean, it's fun. It's an interesting phase of, it is fun. It's exciting. It's a bit crazy, but it gives you stories which many other PIs anywhere in the world may not have. They'll have their own stories, but some of them will not have the kind of stories that we have when you've had to set up, right? I mean, they're adventures.
Right. So it looks like thinking about and tailoring your initial ideas is very important when you are applying for a firm. And so from your initial idea, now your research might have moved a lot, it has evolved and had a particular trajectory on it. Can you tell us a little bit about that transition or that evolution of?
Yeah. So it's interesting. So I'll tell you about two small angles. One was we were very interested in looking at serotonin 2A receptors and whether they're altered by early life stress. And this is one of my earliest graduate students, Madhu's project. And Madhu and I were just sitting one day and talking, and she's like, look, it's not like we are the only ones who've thought about this. There are lots of people who are interested in the 2A receptor, and people had looked at it, but most people had looked at expression, binding, etc. Perhaps to a certain degree signaling, but really that's where it had stopped. It hadn't been looked at from the point of view of behavioral function driven through this receptor. And so Madhu said, I want to try that angle. And some of it is also knowing when to trust your graduate students to totally let them play in a sense, right? So she's like, I'm going to try it. What's the worst? It fails, right? So she comes in and she does the experiment and she stumbled on something really interesting. What she found was that if you went through early trauma, you ended up with significantly enhanced 5-HT2A receptor function behaviorally. We knew this from then further down. We knew this from electrophysiology, other things as well. But the problem was there's no more receptor. And there really doesn't seem to be much. So there's no binding change and there isn't certainly no more transcript or protein being made. So why do you have this enhanced function? And that too in a subset of neurons, not all neurons, but a large subset of prefrontal cortical neurons do show this phenotype. And so then we get stranded, right? Because now it's harder because in those days, there was no single cell sorting in the brain easily in our institute. Then we are stuck. I mean, we've done a microarray, but that's a bash technique where you've looked at the whole PFC. It's not telling you about the granularity of changes. We know it's only a subset of neurons, like 40% of neurons show this phenotype. So when you mush up and you start doing biochemistry, you're going to lose you're just losing the effect. So now you're like, oh God, okay, you found something, but now you're stuck. So you take it further and then you get stuck and now you're stuck for multiple reasons. One, you're stuck because of the access to technology to be able to do the same problem in a particular way. And then you have to kind of figure out a way to diversify out of that and take that same idea. And then so what we did is we went down the road of doing hardcore neuropharmacology. So old-fashioned neuropharmacology, which has its own space. And, we looked at whether blocking this receptor changed behavior, stimulating this receptor in that window changed behavior. And we showed all of that. And that took us, a good solid, this particular project took a fair bit of time. But at every stage, we wanted the granularity and we were getting stuck, right? Yeah. And then, you know that because you attend meetings, you're certainly seeing all the talks and you're going to SFN, your students are traveling, they are seeing it and they're like, oh, it'd be really nice if we can set up chemogenetics in the lab. But to do that. There wasn't anybody in India at that point in time that was actively doing chemogenetics. So it's not like we could pick up the phone and say, can you send us the mouse line? So then you have to import everything from JAX. And then that takes time because, it's just a slow process. So you're getting your animals in, you have to breed them up. And so it took us a lot longer than perhaps it would have taken us in, say, place where you can pick up the phone, call JAX and say, have the animals over by the end of the week.
Yeah.
This took 6 months to just import the breeders, right? Because you were then doing like, DGFT clearance for these things in Delhi and getting all the paperwork done to import your animals. And then you're praying that your animals come safely. And then you're breeding them up. But now, so now when I look back and I think, oh God, that was actually, so there's two ways, right? It is a long game. And so you have to hang in there through that and tell yourself, okay, I know that this will happen. It'll take longer than I think, but I better do a whole bunch of other things in the interim. So which you're doing, but you're also playing the long game in a sense, right? So it takes time. That's all it is. And so then, another graduate student took that up, set up the biogenic mouse lines, took us a lot longer to do a lot of the controls because then we could selectively sort of evoke GQ signaling in CAMK2 alpha forebrain excitatory neurons and then show that when you do that in the postnatal window, you actually program anxiety and program despair and program schizophrenia-like perturbations in sensory motor gating. And we were able to do that. That's what we wanted to do a while back, but it took us longer to get to that goal, right? And I guess the question is, why is it that one can get frustrated along that journey? Because you could say, oh my God, it's not like I had the idea, I know what I want to do, but it's just going to take me a lot longer to do it because I have to do all these other things to get everything in place. But there's also this element of the fact that, when you're setting it all up yourself, and then now people ask us in the country, so it's be a lot easier, for example, now, right? Somebody wants chemogenetic lines, that they're in so many labs in India that anybody can pick up the phone and then you can share. Yeah. So that someone has to do that. And we've benefited from other people doing such things for other techniques and approaches. So at some stage or the other, there will be a little bit of that delay. Now there isn't. Now there just isn't. You pick up the phone and you just ask for things, and reagents are much more easily shared in the country also. So I guess you sign on for that is what I would say. At some level, if you're prepared for it, it doesn't frustrate you so much.
Right. And with just one more question in the same angle. So did the funding that the government provide, or some new scheme and all of those things, does that kind of influence your trajectory, your scientific trajectory as well?
It didn't.
Okay.
Yeah, it didn't. So I had a generous startup funding from the Tata Institute, which the DAE, Department of Atomic Energy. So at the beginning when I came, it's not on the scale of what's given now, but in 2000, it was reasonable enough that I could get off the ground. Came to a very generous department where a lot of senior colleagues just shared equipment. Krishna and Veronica, antibodies, secondary antibodies, half the time my students were scavenging in their fridges and freezers and getting whatever they needed. If I would say to Krishna, and sometimes I don't have access to this, he said, just take it. So there were a lot of people immensely generous, and that culture was quite different from what I had sometimes seen in the US. And I was quite blown away by that. It's rather precious to have that kind of the ease of students going in and out of other people's labs, taking reagents, sharing equipment. So it's much more of a, it's a there's a lot of a feeling of a collective community. So no, I didn't really feel like I had to tailor make my proposals to meet the call necessarily. I went after grants which I thought was suited for what I was applying for. I must say there were grants where I could see the call and I said, there's no way my science will fit this. I would not apply because I didn't want to, I felt like I had an idea I wanted to follow up on. And so I was just going to apply for those grants where I believed that it would be a competitive grant application. Right. And some you get and some you don't get. And that you just sign up for, yeah.
So you talked about when many of the people coming from abroad, they have this anxiety of how do you do things in India? And you have given us a very interesting perspective on that and touched upon some of the unique strengths in India. Can you elaborate a little bit more on that? Like, what are the positive aspects of, like, you heard a lot of negative things, like, how things
I
are
know,
like.
yeah.
So I would like to emphasize on that positive aspects, like, what are the things that you can look forward when you're coming to India?
So I'm a glass is half full person. Okay. So that's the caveat I need to give you at the very beginning. I'll usually look to see, is there something good about whatever, right? So
Yeah.
I would say that when you come back to India, you're coming back for two reasons. Because you want to do research in India. Okay, so there's a research and there's an in India component.
Yes.
Right? Yeah. So now when you say that, if the research component is so much larger than the in India component, you may get frustrated by some of the challenges of the setting it up in the in India system. My advice would be come in because both are almost equally important. Yes, you love research, you want to do it, but you also really care that you want to do it in India, right? So then those two are together and they're fairly equal. Then you're able to navigate when the in India component becomes challenging for some reason. Sometimes it could be getting reagents, getting equipment, not being able to find exactly what you need easily, having to figure out a way around a problem. So those things are just challenges. But, there are different challenges in different countries. So, I mean, imagine you are setting up a lab in the US. I'm sure just getting your R01 funded is going to be a challenge enough as it is. It's not like it's not got, it's just different kinds of challenges, right? So my advice to most people when they're coming back is make sure that the in India component is also really important, because if it is, then you'll manage, you'll figure out a way around, yeah, it'll have your challenges. Everybody has different kinds of challenges, but you'll manage with it, right? I think the advantages are that in most places in India, very rarely do you see everyone in silo structures of departments, right? So for example, let's say you go to an ISER, there will be a whole group of biologists. You come to TIFR, it's a department of biological sciences. Yeah. Whereas you go to a big university in the US or UK or wherever, you will usually say department of neuroscience. And you might even have like a department of neurology, neuroscience, neuropharmacology, neurodevelopment, anatomy, segregated, right? Yeah. This doesn't happen yet in India. And because it doesn't happen yet in India, you end up talking to really interesting colleagues who you might not have talked to so naturally if you were segregated across some huge campus with lots of departments, right? I'll give you an example. Like some of our most recent work is a classic example of it coming up because I talked to a colleague and good friend of mine, right? So so Dr. Ullas Koltur is a biochemist at TIFR, and he's a biochemist that looks at sort of mitochondrial metabolism, longevity, aging, sirtuins, etc. And my lab is very interested in serotonin, the 2A receptor. And we were just kind of working at some interface project where we decided to look at whether serotonin, which is known to have pro-survival effects, has any effects on mitochondria. And we just stumbled on an entire series of observations and then pursued it to its logical conclusion, which showed that serotonin through the 2A receptor versus through a sirtuin and PGC1 alpha axis directly regulates mitochondrial biogenesis, raw scavenging, cell survival, directly influences mitochondrial function. We nailed down the pathway, but, right. I might not have looked at it and he might not have looked at this either if the two of us had not ended up talking about it, right? Now, these things are these interface areas and interface problems, which are sometimes where the really hidden nuggets and interesting things in science lie. Right. And you don't end up, this is what I mean by meandering walks. You have one. So we've been working with 2A receptor for like 20 years now, right? But there's we're largely looking at it from a neuropharmacology, neurobehavior, sort of cellular molecular neuroscience perspective. But then you can also digress and suddenly go into looking at what it does to bioenergetics and mitochondria. Might not have made that digression or wandered off that. Yeah, exactly. So these are the sort of exciting things that can happen just because you end up talking to people whom you otherwise might not have talked to. Nice. Yeah.
With the other question that I would like to see here is like, what are the new topics in neuroscience that are like garnering attention in India now? Like, in your opinion?
Yeah. So, I mean, the neuroscience community is still young in the country. Right. Okay. There was a traditional strength in neuropharmacology. right? So there has always been a reasonably good strength in neuropharmacology in the country, for sure.
Right.
There is also a solid clinical neurology and psychiatry strength of the country. But you know what you didn't often have is crosstalk between the clinicians, either the neurologists or psychiatrists, and the basic scientists who have a PhD and maybe doing more sort of cellular, molecular physiology, like electrophysiology, neuroscience, right? And they're usually working with rodent models. These guys are working with clinical material. Now that in between gap, that was like big valley, right? You were segregated, and some of that is historical, because if you look at the way our campuses are, they're either research institutes or they're science institutes, and the medical schools are completely separate, right? They're not together. It's not like a big university where you have a med school with a lot of people who are MDs, PhDs, MD, PhD combinations. That kind of community is not there that much. I'm beginning to see that changing, right? So that's the really exciting angle that's emerging. And I see this happening in my own work, where now I'm collaborating very closely with colleagues at NIMHANS who have access to, they have the induced pluripotent stem cells that they can differentiate into cortical neurons. They have patient-derived lines that they have the, sort of the genomics angle covered on. So we know individual history. We also know the genome, and we have a sense of what we may know the exome as well. And then we are looking at what we see in rodent models and see if we can translate that, at least in vitro, to cortical neurons that are derived from, human cortical neurons. So that possibility has become available only relatively recently. And a lot of that I will attribute to the India Alliance, which is a DBT and Wellcome Trust funded grant organization, because they have fueled fellowships in clinical as well as sort of basic biomedical, both. And so so you now have an ecosystem that's coming together. And that's really exciting. And I think that will change in the next 10 years more and more so, where you see this crosstalk beginning to happen significantly more. And I think there, India will have a huge advantage, because we have some amazing clinicians in this country, and we have access to clinical material that many other people may not even have access to, right? So there's some stuff here which is incredible that we could do. We need to organize ourselves. We need to create these sort of consortia projects where large number of people work together and build teams that will, it's really team science in a sense that needs to start emerging, but it's beginning to. It's absolutely beginning to. And so that's perhaps in India, the next big exciting thing that's happening, at least in the neuroscience community. Yeah.
And like a final question is, like, as an established faculty, now you are like in the and maybe in the recruitment panel as well. So what would you your advice to early stage investigators who are like, working on their proposals and are trying to apply to the Indian research institutes?
So one piece of advice, you will never have a perfect CV, or at least what you think is a perfect CV. Okay. So please stop worrying about it, because when I talk to people who are applying, they're like, maybe my CV just isn't strong enough. Maybe I don't have as many papers as this institute would like, or maybe I don't have the kind of papers this institute would like. Don't do the second guessing, because you really don't know what the institute is looking for. There is a little bit of a match that has to happen, because institutes have their directions that they want to grow in. And so there is an element of luck. You may be a wonderful scientist, but that may not be exactly what that institute wants to do at that time or grow in that direction. So there's this match that has to happen. So I often say to people when they ask me, I tell them, just don't hesitate so much to apply, because you keep postponing it in this idea of perfection, right? And this perfection is not happening. I don't know what this perfection thing is, right? You can't. So I would say take a risk, take a few risks, start applying earlier than perhaps you think you are ready. Because there is an N+1 problem, which is that you keep saying, I'll do it next year, and then I'll do it next year, and then next year never comes. And what will happen if you apply maximum, you'll get rejected, right? I mean, that's all that'll happen. Nothing more than that is going to happen. So you may meet some interesting people along the way. You may end up, sometimes you go to one location, talk to people, that's not the right fit for you, but they tell you about something else. And it opens up a bunch of other avenues for you. I mean, India has so many interesting institutions to look at today. both in the sort of public space as well as private institutions that are beginning to come up as well. And I would say be willing to take a few leaps of faith. Don't be too hesitant. You know, I mean, you're not, sometimes it's difficult to get everything right.
Yeah.
You know, I think the big things are, am I excited about the colleagues I'm going to get a chance to work with? That's important. Do I feel like this institution will be able to give me the infrastructure, or even if they can't give me the infrastructure, will they let me or facilitate my getting the infrastructure and building the infrastructure, right? So if you get that sense that there is a can-do attitude, there is a desire to do, then pitch in and say that you will be part of the process of making it happen. You're not going to get everything laid out for you. You may have to actually do the work to make it happen. Right. So that's the sort of the big piece of advice. Don't wait for these perfect CVs. I mean, I'm a great example of someone who didn't. I was 29 years old. It was a leap of faith. I took a chance. They took a chance. It happened to work out, right? So it's, if I had told myself, is your CV ready to apply? No, there was no way I was ready to apply. I should have done 2-3 more years of a postdoc, then apply. But I just felt like I wanted to come back to India. I was ready. And they were also ready to say, okay, it's all right. If the person is young still, but they're ready to hit the ground and work, it's fine. And so I think that fear of waiting till the CV is absolutely, you know, where you are satisfied, you may never get satisfied.
Satisfied, yeah. That's true. Thank you, Vidita. This was a really wonderful interview, and I'm sure that, you know, many people have found this, found your journey very inspiring in some sense, and like, you know, that will fuel their transitions to India. Thank you so much.
If there's any way that anybody would like to reach out, I mean, I think people have faculty in India who've just done that maybe a while back, always happy to respond over email or, you know, revert to queries at any time, because it's not, I mean, it's all of us have gone through these events of the process of, you know, you're worrying, oh God, is anybody going to take my application seriously? You know, we've all gone through that. So it's not that it's so distant that anybody's forgotten, right? So we remember it. And when you sit on the reverse side, you also see that, you know, it is a challenge to figure out where in the country. There's so many new institutions now, but that's a good thing. There are so many more options. Right?